This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 268, recorded on January 17th, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Nice weather today, isn't it?
Yes, it's 4.5 degrees Celsius right outside my garage, 51% relative humidity, and a barometric pressure of 1,015.2 millibars and falling.
Right? Why is it right outside your garage? You got a sensor there or something?
I did. It's going to be my pick this week.
Nice. Yeah. And you can see it from where you are?
Yes, I brought the little indoor console that it just sits like a picture frame. And
cool.
Got it sitting on my desk right now.
Yeah, we're 7 degrees C with partly cloudy skies. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
How you doing?
Okay, I'm trying to figure out my weather. It's not coming up on my computer dashboard, but it's 29 Fahrenheit.
It's colder, below freezing. Yeah,
it's not coming up in Celsius, so you just have to do the conversion. Sorry.
It's all right, but I can tell it's below freezing.
And we had sun about a half hour ago, but it's gone away now. But it's still pretty nice.
The sun is about to set over the Hudson River, maybe another hour, I would guess. Right here in the TWiV studio, we have a return guest from right here at Columbia University, Ashlee Bennett.
Hey, Vince.
Welcome back.
Yeah, I'd say the weather over here on my side of your office is probably a nice and balmy, maybe 73 degrees.
Yeah, you wish. It sounds like island weather. Actually, I just interviewed a student from Berkeley, and he said it's in the 70s there.
Oh.
Do you remember what TWiV you were on last?
Wow, it's been over a year, so it's been a while.
We do 50 a year, so it was 200-something. Anyway, Ashlee is here because Ashlee is the TA, my intrepid TA for my Coursera virology course, and also for the real Columbia. I shouldn't say real, right, because they're both real. But the physical Columbia course. And as part of the Coursera course, which began last week, right?
Yes, the second semester began last week.
Ashlee thought to involve the students and talk about it just for a few minutes on TWiV. So why don't you tell us what you did, Ashlee?
Well, this semester we wanted to try to engage the students in more of a discussion that similar to what you would get out of the in-classroom experience, where a conversation might start during the class or after the class about something relevant to the course material and day-to-day life. So we started this segment called Join the Discussion, where we post a discussion topic question, and students can go in and submit their thoughts on it. And to encourage the non-mandatory participation, we said that we would give shout-outs to the people whose posts we mention on TWiV. And, that's a huge incentive because TWiV is so wildly popular, and it's how they can get their 5 minutes of nerdy fame.
So did you have a good response last week?
Yeah, I would say we had a pretty good response, especially since the participation is usually lower in the first week of because not everyone is enrolled yet. But it was a pretty good response. Quite a few responses to sift through. So
you're gonna basically post every week some discussion topic, and then we'll pick a couple of good ones, or you'll pick a couple of good responses, and you'll read them here. Is that the idea?
Yeah.
Okay. So what was last week's discussion topic? So
the discussion topics are always loosely based upon that week's material. And last week we were learning all about the basics of viral transmission and disease transmission in general, such as vertical transmission, horizontal transmission, nosocomial transmission. And one of the things I wanted to get the students talking about was the idea of fomites in the doctor's office, because there have been many studies that have come out in the past few years about how doctors' ties or their purses can be a fomite, which is something that can be used to transfer a pathogen from one person to another. And I wanted to see what students thought about this.
It's a good one.
So I'll just read the prompt that the students got. It's called Fomites in the Doctor's Office. Should regulations be in place based upon what we know? Iatrogenic and nosocomial transmission are a huge problem when it comes to controlling the spread of disease. That's why they have their own special vocabulary words. In recent years, numerous studies have shown that seemingly innocuous practices, such as having magazines or toys in the waiting room and doctors wearing ties or carrying purses, can aid in the exposure of microorganisms to patients because these objects can act as fomites. However, despite these studies, many clinicians continue with these practices even as the threat of hospital-derived antibiotic-resistant infections increases. Should such practices be banned? Should the professional attire of physicians be dictated by the results of these studies? And should magazines and toys in the waiting room be a thing of the past?
As I said the other day, we should have stainless steel waiting rooms with nothing in them.
As soon as you walk in, you have to strip and shower.
Great.
Yeah, disposable gowns and masks for everybody.
Well, I think this topic is really interesting because I also remember a study that came out a couple years ago showing that patients had a sort of predisposition towards doctors who were professionally dressed. So they were more likely to consider a physician wearing a suit and a tie to be very professional and very smart and knowledgeable over one that was maybe, chilling out in scrubs or some other more relaxed attire. So I think it's important because we may need to have our perceptions changed on what is the appropriate clothing for people in these positions.
Well, at least with respect to ties, they recommend bow ties, but to some extent, for me, that always creates a bit of a comical image.
Yeah.
Yeah.
Well, and there have been a number of studies that find that people are considered less trustworthy in a bow tie.
Oh, really?
Is that right?
Yeah.
It kind of reminds me of like a used car salesman, I think.
Yeah, exactly. It's
fine.
Or
it's
a clown.
comedic
Yeah.
So what kinds of responses did you get?
Well, there were a few main categories. I did get a series of responses from people who are probably self-identify as libertarians, who were like, no more regulations. Doctors shouldn't have any regulations. Too many regulations. I'm not really going to go into those. Some of the other main categories were there were people who thought that we should take these into consideration. And I'm just going to read one of those. This is a post by someone named W. Clifton Baldwin. He says, toys and magazines in the waiting room are tradition, but tradition is not a reasonable argument for keeping them. When I would go to the doctor's office as a child in the 1980s, there would be at least an hour in the waiting room from the time of the appointment until actually being called. Although my doctors are different, I rarely wait longer than 10 minutes these days in any doctor's waiting rooms. It is not their responsibility to provide entertainment. Furthermore, most people in the waiting room are looking at their phones or some other device they have brought anyway. Similarly, for children, parents can, and appear to do so from my limited observations, bring something to amuse their children. When all else fails, most doctors' waiting rooms have televisions that can provide temporary entertainment. Therefore, I do not see a good argument for having magazines and toys in the waiting room. Since research has shown that toys and magazines can spread disease in medical settings, there is support to eliminate them. I understand that it will not completely solve the problem, but it should reduce transmission somewhat. Hence, I see no strong argument to keep toys and magazines, but research has shown why they should be taken out of the medical setting. So there were a lot of posts of that nature, basically agreeing that given the research we have, the data, that we should modify our clinical practices for this.
Okay.
The second type of response I received was, and I don't mean it to sound bad, but it's sort of there's so many different ways of being exposed and in things being transmissible around the doctor's office that we should just essentially give up. And so this is the Jocelyn Perez posted. For this discussion, we are considering ridding fomites such as toys and magazines and doctors' personal items, like ties and bags. But isn't the office just a cesspool for disease in itself? The seats themselves, the floors, the doorknobs, water fountain, public restroom, and even the counter and sign-in sheet, and don't forget the pen. I understand the sense of urgency in regards to toys and magazines, but all these other forms of transmission are present. Even the air is teeming with pathogens. If we were to agree to do away with toys and magazines, ties and handbags, we might as well do away with seating in waiting rooms and just have a room that is continuously being sterilized. Even the doctors, nurses, and equipment such as the scales are potential hazards. We sadly cannot escape exposure in every regard. Would removing toys and magazines lessen your chances? Probably. However, it will not protect you from catching something. Transmission is inevitable.
I like that.
Nice.
I like the use of cesspool. It kind of made me think differently when I went to the doctor earlier this week.
The world's a cesspool. You know, I pass a pediatrician's office on the way home, and he has two doors. One says well patients, and the other says sick patients. So I presume they go to different rooms, right? Mm-hmm. So that's one way. Because, you know, they're a pediatrician's office, you often have sick kids and well ones all thrown in. Anything else?
So the third type of response was one where it recognized that taking certain steps is not going to, be a cure-all. It's not going to prevent any types of transmission from occurring. But anything we can do would be helpful. So Lucia posted, "Hi, I think it's important to restrict the use of toys in every kind of fomite because obviously in hospitals and waiting rooms where there are sick people, there are many microorganisms in the environment and in all these toys which may be innocuous for some people, but in sick kids or in immunocompetent patients, it could be fatal. I think that to reduce these toys should be considered a practice to protect these vulnerable individuals." So I think that is an interesting point. I didn't think about the immunocompromised. And so we had a couple other responses that were to that effect that perhaps even though it's not a cure-all, it would be a good idea.
Any other kinds of responses, or basically those three kinds?
There was another responder named Cornea Casey who suggests that we should treat all doctors' offices and waiting rooms like BSL-2 facilities.
Like our lab, right?
Yes, like our lab, which I don't even know if we're that strict about anymore. But I thought that was an interesting idea as well.
That's kind of already the case. I mean, the waste stream from a doctor's office is handled specially. I don't know that it's regulated in quite the same way, but there are rules about containment that relate to doctors' offices.
Yeah, but you don't have to gown up and glove up.
No, you don't have to gown up and glove up. It's not quite as stringent, but there are regulations along those lines. Personally, I think if you're going to focus on something like that, it should be getting physicians to wash their hands more often.
Yeah.
Yeah.
So the other thing I learned from these posts is that apparently doctors have been paying attention to this data. And there's a post by someone named Elizabeth Lambert, and she says, "My children's pediatrician does put away all toys and books during flu season. It's nice to know they take extra precautions to keep our children well." So that was something that I didn't know, that some doctors do take that into consideration. So I guess the research, it's not falling upon deaf ears, but yeah.
Well, in the future, everyone will bring in their tablet and just read that, right?
Yeah, well, speaking of touch screens, one last
which
thing.
Which is its own fomite, yes.
So Peter John Foster posted, "My doctor's surgery uses a touchscreen computer for patients to book in when they arrive for their appointment. I often wonder if the screen is ever tested for pathogens." So to that effect, I did some traveling over the Christmas break, and I noticed something and thought about it in a way that I guess reveals the fact that I've been trained to be a microbiologist. I was in the airport, and they had these areas kind of like in the waiting section of terminals where there were iPads with touch screens on, kind of like attached to this kiosk on a table that anyone could go up and touch. And this is in an airport. And so I just thought to myself, what a fantastic way to spread diseases around the globe. Some guy this is in a New York City airport, some guy can fly in from Beijing, he touches that touch screen, and then another person on their way en route to Tennessee touches the same spot. Next thing you know, you could get an exotic disease showing up in Tennessee.
Ashlee, we used to have these things called telephones in airports, and they were public phones, and people used to put their mouth and ear right up to it.
Oh, yeah.
And then the next person, and as far as I know, Well, they're gone now, of course, because everyone has their own private. So this seems less troublesome than those. I don't know.
Yeah, probably.
I'm trying to find something, but I just saw something in the news where somebody had done a study about microbes on tablets and phones and how people really probably aren't cleaning their screens as often as they should.
Well,
So, but I can't find that. Yeah.
When it's your personal tablet or your personal phone, it's probably less urgent to do so than a community device. I also went to the Columbus Circle Mall recently, and I was looking for a store, and instead of having like the old school style directory where you just like look at it until you find out where you and like use your map skills to figure out where you're at, they had a touchscreen system where you go up and you like touch it and you choose different options until it tells you where you want to go. And that was another case where I just felt like it was sort of like unnecessary touching. I think touchscreens are great.
But you rode the subway there and you held onto the bar while the train was moving, didn't you?
I did, but I usually try not to make bare hand contact if I can. I'm a very, very talented leaner.
Do you wear gloves all year round?
I don't wear gloves all the year round, but oftentimes I might just wrap my arm around the pole, or I try to make sure it was just one hand touching. I mean, you're always going to have to touch something, but you can try to limit the amount of things you were touching.
Well,
I just washed my hands after riding the subway.
Yeah, that's it. The key is if you are in a public place and you touch a public thing, like a tablet or a pole, you just don't touch your mouth or eyes before you wash your hands, right? Mm-hmm. But unfortunately, most people either don't or don't have the opportunity.
Yeah.
I don't know. It seems to me that, there are always ways to get infected, and if you take some away, others come to take their places, and I'm not sure we can control it at all. Maybe that's the moral from this interesting episode.
It's a Sisyphean task.
Yeah.
I have a question, Vincent. So this is your Virology 2 class? So this is a continuation of the other one. Is that correct?
It's technically a separate occurrence on Coursera, right? Because the first my class is too long for Coursera, so we broke it up into 2 courses. So it's basically Virology
part
2, but you don't have to have taken part 1 to take part 2.
And then will there be a time again when you offer Virology part 1 again?
Yeah, if I want, I could do it again next summer or this summer. Yeah.
Just curious.
Yeah, it'd be interesting. We had 26,000. Is that right for the first one?
Yeah.
And we have 14,000 for this one. I'd be curious to see if we repeat them, whether the numbers keep going down and down and down, or whether we just have new people every year who are interested. Mm-hmm. That would be an interesting experiment. So you're going to come back next week, Ashlee?
Yes, I will come back next week with our next discussion question.
Do you have one made up yet?
It involves inflammation.
Okay, cool.
So,
of course,
it should be a hot topic.
Inflammation.
But ching.
Well, thank you, Ashlee.
Thank you.
See you next week. Well, I'll see you sooner than that, but
yeah. Okay.
All right. Bye.
Bye.
Take care.
Ashlee.
All right. That was Ashlee's idea. I thought it was a good idea.
Yeah.
And of course, if TWiV listeners want to weigh in, you know how to do that. TWiV at TWiV.tv. We have a follow-up from Jessica, who writes, "Hi, TWiV gang. Since you invited people to send in their do-it-yourself viruses, here are mine." She's knitted an icosahedron with 20 faces, 12 faces, vertices. Well, 20 faces, 12 vertices, right? And 5-fold symmetry. I made these many years ago to give to my graduate advisor, Michelle Osmond, whom you have had on the show. Thanks for the great podcast. Yeah, so for some reason this time I had to log into Ravelry. Last week we didn't have to.
We didn't have to. I think she gave us last week the direct link. I don't know what was different, but this website is not a commercial website. It's they will never spam you, according to my friend Sarah. It's run by, I think she said, the spouse of one of the fiber arts people, knitters or whatever. And so they're very happy. It took me a minute to log on, get a username, and then see whatever I wanted to. So
yeah, I just made an account, and so I got her pictures, and we'll make them the image of the week there up on the TWiV post. So I got curious. I said, what else is on Ravelry? And if you just search for virus, you get about a dozen people who have made fiber arts of viruses. So I think I'll put a link to that in the show notes. So thank you. Anyone else has fiber viruses, DIY fiber viruses, send them in. We'd love to highlight them.
I guess you could rub some grease on one of these and make it an envelope virus, right?
That's right. An envelope is a challenge.
Or just put it in a manila envelope.
Yes,
There you go.
yes.
I got an email from Samuel who writes, together with Roland Regos, we're organizing a conference on viruses we thought your audience might be interested in. Our aim is to bring together people working on different viruses using a variety of approaches. For instance, we invited people like Jean-Michel Claverie, who works on giant viruses, and Christophe Fraser, who is an epidemiologist. So they would like a little bit of advertising, and happy to do that. I will post the information for this meeting in the show notes. It's going to happen April 2nd and through 6th in Roscoff, which is in Brittany, France. It looks like a good lineup of people.
Then, hey, if you guys need somebody to cover that conference, let me know.
400 euros for PhD students, 600 euros for others.
Okay.
That's for the meeting. So check that out. We're happy to advertise your meeting, by the way.
Mm-hmm.
Yeah.
And it's always nice if you advertise TWiV. I'm losing my voice because I just interviewed 3 students. Okay, this is... Now we go on to papers. We have 2 papers today. And the first one is going to amaze everyone. This is actually a paper that Kathy sent. First, because it's a plant virus, which we hardly do, right? Mm-hmm. And second, because the mechanism is really, really cool. And this, I guess it goes to show that we need to do more plant viruses, because I was totally blown away because I didn't know any of this. I don't know if you guys knew it all.
No, I didn't know this story either.
I didn't know this at all.
So the virus in this paper, it's a Journal of Virology paper, and it's entitled Virus Factories of Cauliflower Mosaic Virus are virion reservoirs that actively engage actively in vector transmission. And the authors are Bach, Gargani, Masia, Malouvet, Verneret, Blanc, and Drucker. And they're from... all from France, right? Montpellier.
And they have a related paper from beginning of 2013 in eLife, which is the main one that I read because I asked Vincent for a copy of the JV one and he didn't get around to sending it to me. But
I did.
You did?
Yeah, I did. You didn't get it?
I didn't get it.
Sorry.
Didn't come through. That's all right.
Yeah, the eLife is quite good.
The eLife paper is open access and has some very nice data in it as well.
Yeah, and the figures in the eLife are pretty cool. I pasted some of those into the show notes. So,
and I love this thing they do on eLife. I hadn't noticed this before, where they do right underneath the figure, you have the figure legend and then the source data.
Yeah, yeah.
And you can, if you want to, you could download any of the data that went into producing that figure. I think that's a really cool idea.
Mm-hmm.
Yeah. So a cauliflower mosaic virus is a virus with a double-stranded DNA genome that also encodes a reverse transcriptase. So it's strange. It's kind of like hepatitis B virus, in that the virus packages a DNA genome, but it's a reverse transcriptase encoding virus, and that's because the virus actually encapsulates RNA in the cell, and retroviruses produce particles with RNA, but these cauliflower mosaic viruses and hepatitis B viruses reverse transcribe before the particles get out of the cell. They can't wait. They can't wait to start reverse transcribing. So they have DNA in the particle. These viruses infect cauliflower, as you might expect, and also turnips, and they make them sick. They cause a variety of symptoms like lesions on the surfaces, stunted growth, and deformation. And these viruses are transmitted by aphids. So there's a good discussion and introduction in this paper where they point out that, in fact, most viruses are probably transmitted by vectors and not by the way humans transmit viruses on subway poles and doctor's office toys.
Right, because plants don't ride the subway.
Plants do not. They don't seem to go to doctors either.
No.
They probably don't go to the opera.
And too bad they're missing out, right?
Right.
Although, not Wagner. So they're transmitted by aphids, and then they give this in the eLife paper. There's a great, I think it's in the eLife paper. There's a great description of how this happens.
Yeah.
Before we get to that, so in an infected cell, a cell infected with this cauliflower mosaic virus, there are two types of what we call inclusion bodies. These are kind of assemblies where virus particles are being made. I think we've talked about viral factories in terms of pox virus replication. It's just a way of concentrating all the components that you need. So cauliflower mosaic virus infected cells, they have virus factories and they have transmission bodies. And the factories are, I think, thought to be a place where the virions are assembled, and it was previously not known the fate of those virions. But the transmission body, which is a different and distinct inclusion body in the cell, is required for transmission, as the name would suggest. And in order to get transmission, which again occurs through an aphid vector, you need to have a complex formed, which is composed of the particle. The particle has a shell made of a protein called P4. Then there's a couple of other viral proteins called P3 and P2. And P2 is quite interesting because, so this is a capsid protein, or it's a protein on the outside of the capsid that links the virus to the stylets of the aphid. Now, Alan, what would you say a stylet is?
It's kind of like the aphid's lips.
Mouth parts?
I mean, they're not technically the lips, but the aphids feed by sucking sap out of plants. So
Right.
they have these sucking mouth parts that they stick in, kind of like a mosquito. And at the very, very tip of this, there's this stylet, very fine thing. There's a picture pasted into the show notes, which I think is from the JV paper. And at the tip of that is where the virus attaches. So there's this. The eLife paper also has a nice schematic diagram of this, where you've got the tube going up through the stylet where the food is going to go, and there's another tube coming down it where the salivary gland sends out saliva. So the virus attaches right at the very outer edge of this whole stylet.
Right. It's amazing, right? It specifically attaches.
It specifically attaches to a receptor that's found at the tip of the stylet. So it's going to be right there at the tip when the aphid bites the next plant.
Right. And they actually call this P2 aphid transmission factor or helper component. I think they kind of distinguish it from, they don't really consider it a virion protein. P3 and P4 are, at least in this one figure, part of the capsid. But this thing is like a little extra thing.
Right, I gather
hanging off.
you don't need P2 to make a complete virus capsid.
Right.
You can make the virus capsid with P4 and P3, and then P2 seems to just be there to link
Right.
the virus to this aphid receptor so that the aphid doesn't swallow the virus.
So they say in this paper when aphids feed on plants, they insert their stylet into the space between cells, and then they do what they call a series of brief test punctures. So that the aphid is looking for a continuous source of sap.
This is a common thing with sucking insects.
Is that right?
Mosquitoes do this too. They probe around under the skin until they hit a blood vessel that they can drink from.
Right. And so they say the plant is okay with this.
Not that it can do anything.
Right. Because only a little bit of cytoplasm is ingested. And that's the way this virus and many, many other viruses are acquired. And they get into the aphid. Now, this is kind of an interesting background. So some viruses get into the vector and they're ingested, and they will get into their gut and sometimes replicate. But in other cases, they just attach to the vector. Like here, the virus attaches to the stylet tip. And then when the aphid leaves, the virus goes with it, and it brings it to another plant. And then the virus is injected as the aphid probes another plant. And I don't know, you were at ASV, Kathy, you heard it talk about similar things with different vectors and hosts, right?
Mm-hmm. Mm-hmm.
I think it was a nematode or something like that. Mm-hmm. Now, the cool thing here, so that's how the virus is taken up by the aphid, but the cool thing here, which is really the topic of the eLife paper, is at the precise time when the stylet penetrates the host, somehow the infected, now there's going to be an infected cell in some hosts, right? Mm-hmm. Somehow the virus-infected cell senses the aphid and produces a transmissible form of the virus so that the insect can take it up. And what happens is that we have these transmissible bodies in the cytoplasm of the infected cell. Tubulin comes into it. The P2 protein relocalizes onto microtubules throughout the cell, and they form, the virions then associate with these P2 decorated microtubules, and they form what are called mixed networks. So basically you can imagine these networks of microtubules now that are lined with virus particles. And that, I guess, makes it easier for, or more likely that the aphid is going to take it up, right? Mm-hmm.
Right, and the virus actually seems to move from the virus factories into this new network of the if you look at the cell before the aphid bites, you've got the transmission body, and it has maybe a couple of viruses in it, and then the virus factories are where most of the virus is hanging out.
Right.
And then when this transition occurs and the transmission body reorganizes, then the viruses from the virus factories that are all complete go rushing into this new network, this new microtubule network in the cell.
Right. And that's the question in the JV paper. Where did they actually show that the virus comes from the virus factory to the body and eventually to the aphid. The cool thing is when the aphid leaves and there's no more stress on the cell, everything reverses.
Right.
Mm-hmm. The viruses come off the microtubules and new transmission bodies are made. So ready for the next aphid. It's just amazing.
Yeah. So this was the cover photograph for the issue of Journal of Virology, and the cover photograph and the caption are open access. So I think you should put the link to that in the show notes.
Right.
Because
very cool.
The green aphid, it's a composite photo. They have the green aphid scanning EM, and then they have these different purple cells with the different forms of the inclusion bodies. So
it's really neat. So again, the aphid feeding induces this change in the cell. So I mean, that's something that's not answered. How that is sensed, right? How the aphid is sensed, and how that is transmitted to make the structural change. That we don't know, and that would be really interesting to know, of course.
Right. They just say it's through host factors, presumably.
Yeah, and then they did some math,
Right.
Yeah, the picture is so cool.
Yeah.
I think I remember seeing that. So the JV paper is a little anticlimactic because that's the coolest part of this. But it's work that had to be done to figure out where the virus is coming from, right? That goes into these transmission bodies. And the paper is a lot of microscopy using antibodies to different cellular and viral proteins.
It's beautiful microscopy. Both the fluorescence microscopy and the EMs.
Right, so they do both, gorgeous. Yeah. So fluorescence to get a picture, and then when they want to get some details, EM. So they show initially, and this is quite interesting, that they, instead of having to have an aphid feed on infected cells, they just expose them to carbon dioxide or to azide. And this somehow mimics the signal so that the virions then jump to the transmission bodies and relocalize to the microtubules to form these so-called mixed networks.
Right, different types, and it seems like a variety of types of stress cause this response.
Yeah, so that's, I guess, the probing with the stylus or style, what is it called? Stylet. I guess it's a small stylus, right? The probing is stressing, and so CO2 stresses and any kind of stress. Yeah, that's probably it. So it's a stress response, I suppose.
Yeah.
So there's some clues.
It's worth pointing out that in the very first experiment, they use infected leaf tissue.
Yeah.
But then they go on and do all their subsequent experiments using plant protoplasts. And so for the listeners that might not know, the plant cell walls have been removed when you make protoplasts. In this case, they digested them with cellulase and macerozyme, 2 different enzymes. And then oftentimes, they don't do it in this paper, but you can take protoplasts, do things with them, and then you can regenerate the whole plant from those protoplasts.
Right.
Again, it's not something they had to do in this paper, but...
Did you work on a plant virus, Kathy?
I came close when I was changing advisors. Working in Steve Howell's lab was a possibility. So a lot of my fellow graduate students worked with cauliflower mosaic virus.
So I guess in a medical school, it's pretty rare to find plant virology, right?
Right.
Yeah.
Although there are always exceptions, but it's hard to... Your colleagues just don't understand what you're doing.
Right.
If you're there.
Right.
So I think the first part of the paper, I guess the first figures, they're showing that this CO2 stress basically leads to the formation of these mixed networks. So viruses associate with microtubules, and these are connected among each other. They're mixed networks, so they're all connected to each other. And then they do a cool set of experiments where they use a virus lacking the P2 gene. All right, so this is one of these protein. The P2 is the protein that links the virion to the stylus. Is that right?
Stylet.
Stylet. Never going to get that right, you know. The P2 links the virus to the stylet, so it's absolutely required for transmission. If you take the gene out, the virus will replicate, but it would just not be transmitted. So they established this, I think, in protoplasts. I don't remember.
Everything from here on is in protoplasts.
So it's in protoplasts now. They infect with this deletion mutant, and these virus-infected cells, they have virion factories, but they don't have transmission bodies because you need a P2 for that. And then they express, they produce the P2 protein. Hey, I caught it. They produce the P2 protein from a plasmid in the infected cells. And in fact, when they stress these, they form transmission bodies, and the virions go into them. So this is a cool system that you can now use to manipulate. By having the P2 separate, you can look at the temporal formation of the transmission bodies, and then you can start to get an idea where the virions are coming from.
And then this is the first time where they do an electron micrograph, and I was just fascinated by this because they talked about electron-lucent parts of the micrograph, both in this figure and the next one. And it came up because a student in class the other day wanted to know, what does electron-dense mean?
Yeah,
and I'd never had to explain that before because it just seemed obvious to me.
Yeah.
But electron-lucent is where there's less staining, less binding of the stain. So
electrons are passing through, right?
Mm-hmm.
Actually, I forgot to mention that in one of the figures, they showed actually virions by EM lining up along microtubules, which of course is what happens when you stress the cells so that they're getting ready to be picked up by aphids. So they do a kinetic experiment where they put P2 at different times after infection, and from that they can show that it's most likely that the virions originate in the viral factories. Because one of the ideas, I think, was that P2 is dragging the virions from the factories to the transmission bodies, and by expressing the P2 separately, they could show that that wasn't the case. Mm-hmm. Let's see. Next set of experiments, they're using azide to stress them.
Because I thought the pictures were so fascinating, I did what my first thesis advisor said: a good paper, you should be able to
look at the figures,
read the abstract, look at the figures, and then figure out the paper. Yeah. So this figure is really just showing that the azide stress makes the virus release from the virus factories.
Right.
And they see these paracrystalline arrays, but they only see those with the mutant virus.
And they also show that they find some virion factories where they seem to find virions budding from the surface of the factory, which would be maybe how they get out of the factory to to get into the transmission body.
Right.
And I think they also show in this experiment that these virions that are released from the factories by azide can be picked up by aphid vectors. So again, the system where there's a mutant virus lacking the P2 gene, where it's expressed ectopically, they stress the infected cells and let aphids feed on them, and they can pick it up and move it to other cells.
Yeah, that's pretty cool. They can just have these infected protoplasts, evidently, in solution, and then put the aphids on it, and they suck it up, and then they put the aphids on plants.
Right. Then they did an experiment where they actually put a epitope tag onto P2 so they could more closely follow it during infection.
Well, so in this figure, they're trying to show that the mutant virus is actually recruiting the virions. That's a weird title for the figure. Recruiting virions from the virus factories. That in a virus infection with this mutant, the virions are being recruited from the virus factories to these microtubules.
Yeah, so they can actually look for P2 and show that it's not in the factory itself.
Right.
It's in the transmission body. So again, the idea is that it's not pulling virus out of the viral factory.
Right. In fact, this mutant virus doesn't even make the transmission bodies.
Unless you
so
express,
that allows
yeah.
them, yeah. So that allows them to look at this more carefully.
Yeah. So you can, but you can make them if you express the P2, right?
Right. Right.
And then the next experiment, they show that if you take CO2 away, if you remove the stress, then the mixed networks disappear, and the viruses go back into the virus factory, the virion factories.
Mm-hmm.
It's just
you
amazing.
missed the aphid, go back to the waiting room.
That's right. That's right. Or maybe your buddy got picked up and you can wait till the next train.
Right.
And they see in a number of cells what they call virion aggregates, which they suggest may be part of the mechanism for these viruses to get back into the virus factories.
Mm-hmm.
They also do an experiment, right, where they add exogenous virions to these cells. They say that we transfected fluorescent virions into protoplasts.
Mm-hmm.
It's funny that you would transfect a virion. And then they can follow these getting into the virus factories after withdrawal of the shock. So that's pretty cool, too.
Yeah, so those added exogenously definitely co-localize with the virus factories. There was some question about, if they're just free in the cytoplasm, will they go back to the factories? And that's their evidence for that.
Now, my guess is that what this is doing for the virus, I mean, the question would come up, why not just leave the transmission body in its, ready to transmit state all the time? But presumably that would damage the cell, right?
I don't know if the cell's microtubules are lined with particles. Yeah, maybe that's a problem.
So maybe maybe this is a mechanism for letting the cell survive, which obviously the virus needs to have happen.
Yeah. I like that.
And then get ready when the aphid arrives.
So I think the thing to remember here is that as these cells make viruses in the viral factories, that's the end of the line unless they're picked up by an aphid, right? Mm-hmm. So, I mean, in terms of transmission, they could spread within the plant, I presume, right? And there are mechanisms for that. But so they sit in the VF until an aphid probes, and then they hop onto the subway.
Yeah,
it's really cool. I wonder if they touch the poles.
Well, they stick to
the poles. Yeah,
they do. They grab on.
Yeah, the stylet pole.
So as I said earlier, I really think the signal here, I mean, as we said, is probably stress triggered by the aphid, and I think that would be interesting to look at.
Yeah.
Mm-hmm. They also have a nice discussion point where they talk about the particles are important for getting the virus to the nucleus in the first place.
Right.
And then they are important for the cell-to-cell and movement within the plant.
Yeah, yeah.
And then they're important for getting to the next host by way of the aphids.
Right.
So it's a lot of stuff that has to happen.
Yeah, they make the neat point that maybe the virions are kept in virion factories to prevent them from reinfecting the nucleus. Mm-hmm. So once they're made, you don't want them to go back in the nucleus of the same cell. So how do you prevent that? Maybe by putting them in a virion factory until which time that you have to be transmitted.
Yeah.
Anyway, so it's cool.
And, I guess, thinking about it, maybe the aphid stylet is not going to have as high a probability of hitting a transmission body as this microtubule network.
Right.
And the transmission body goes away, and the viruses are now in the microtubules, and I don't know.
Yeah. It's
just beautiful microscope work, and a couple of really nice models to look at, both in the eLife paper and then this cover image from JVI. So
very cool story.
Yeah, so plant virology is very cool, and there's a lot of different stuff that has to happen, and I think we should do more of them.
Mm-hmm. And it's a very important. I mean, we think of human viruses as being more important, but if the plants go away, we're gone too.
Yes, cauliflower mosaic virus is one of the top 10 viruses in plant pathology.
Yeah. This is a huge agricultural problem, right? Because this thing is very common. It's aphid transmitted, and the aphids are everywhere, and farmers have to deal with it all the time.
Yep.
All right, our second paper is a science paper called Comparative Analysis of Bat Genomes Provides Insight into the Evolution of Flight and Immunity. A lot of authors on this paper, and I'm not going to read them all. But they are from, some of them are from Australia, some of them are from Copenhagen, some of them are from China, Maryland, Singapore, Denmark. Oh, did I say Copenhagen? Yeah. Anyway, so this is a paper where they sequenced the genomes of two bat species, the black flying fox, Pteropus electo, and David's myotis davidi. I love it.
David's myotis sounds like somebody's disease.
Yeah, myotis, little bats. And these are two types of bat that have been the genome sequences were done on captive or captured bats, but these bats are used for scientific research. There are colonies of them. There are cell lines. There are reagents for these bats, and so this is the first time that they have sequenced their genomes.
And these
I was going to point out that they're from two different suborders.
Yes.
And they are the yin and yang suborders.
Yes.
Yinpterochiroptera and the Yangochiroptera.
Yes.
Cool.
And they are that seems appropriate because they're very different. One hibernates, the other doesn't. One eats fruit, the other eats insects. So one is echolocating, the other is not. So they're very distant relatives.
So this paper actually was suggested by a reader. I think we mentioned it last week, and also Rich had brought it up in response to a listener question last week. So they want to know if they could get any clues about two major bat issues, and one is how they fly so much. They're always flying, and that requires a lot of metabolism, which could produce reactive oxygen species that would damage DNA. So how did the bats deal with that? And the other, of course, is how do they deal with so many virus infections? We know from many TWiVs that bats harbor many different kinds of viruses. And so can you deduce something about their ability to do that from looking at the genome? So they sequenced the entire genome at 100x coverage. That means each base was read 100 times, at least, on average. The genome is 2 gigabases. I'm so biased for my computers. 2 gigabytes, 2 gigabases long, which is a bit smaller than other mammals, but has 21,000-ish genes, which I guess they identify by various criteria, including open reading frames. And one thing I found cool is that one of the species, Electo, has an endogenous herpes virus in the genome. There are 126 copies of an endogenous Cymerine herpes virus 2. Isn't that cool?
Yeah.
So I guess maybe it's doing something, right? Because it's expanded and maintained. Who knows? So they looked at all the genes and tried to figure out if they could get any clues from looking at them. And so one of the sets of genes has to do with DNA damage. And they find that a lot of them are under positive selection, which means that the non-synonymous to synonymous ratio is greater than 1, right? Did I get that right? Non-synonymous, which means makes it an amino acid change. So
right.
DN to DS is greater than 1.
Yes, so there are more amino acid changes than changes that are not amino acid changes.
Right.
Yeah.
So that would suggest that there's been some evolutionary pressure on these genes to make them change. And in fact, they look at a number of genes in the DNA damage checkpoint repair pathway. So there's this really cool pathway where if you get DNA damage by whatever mechanism, it can be sensed by proteins, and then those proteins can go on and induce DNA repair or halting the cell cycle so that you wait until the repair is done, or can induce apoptosis and kill the cell. So proteins like ATM and DNA protein kinase and BRCA2 is one part of this DNA repair. P53 is another member of the pathway. So they all seem to be positively selected, which they say is good because if these guys are flying a lot, that would be consistent with dealing with the DNA damage, right?
Yeah, flying requires very, very high metabolism for an animal, and that's going to generate a lot of reactive oxygen, and so this could confirm their hypothesis that it fits. It's consistent.
I don't know how they would test it, though, right?
Yeah, that's the problem. You need a non-flying bat to control.
Maybe you could do something in a cell line. You could knock some of these out and apply oxidative stress in some way. I'm sure they're thinking about it. Mm-hmm. And they also looked at immune genes, genes of the innate immune system, and they also find positively selected genes there. A number, the CREL, which is part of the NF-kappa B pathway, is positively selected. And CREL is interesting because it's also part of the DNA damage checkpoint pathway. Yeah. But there are other, toll-like receptors and interferons and interferon components, ISG15, which we talked about in Austin, the little ubiquitin-like modifier. So things that are involved in defense seem to be positively selected. And I guess the point here is that these are positively selected, whereas other genes are not, right?
Right.
Mm-hmm.
Which would make these special. Also some differences in NK cells, natural killer cells, which recognize infected cells. They seem to have some alterations, some differences from other mammals, which suggest that they may use different ways to recognize infected cells, which could be part of why they're so good at handling virus infections.
Right. And it also turns out that they lost a locus, the PYHIN gene family, which is a microbial DNA sensor.
That's right, yeah.
And that's weird because everybody else, all the other mammals, have that.
Mm-hmm.
And their theory is that because these genes are probably involved in both DNA damage and antimicrobial response, their DNA damage role, they had to get rid of them in order to have the high metabolism. Mm-hmm. And so this is the price of flying. You had to get rid of this mechanism that was getting in the way of the high metabolism you needed, which could, it fits in with a model where bats are not only the only flying mammals, they're also the ones that seem to have the most viruses.
Mm-hmm. Right. And I think that's basically the story here, right? Anything else?
Right. So did we not have any bat genome sequences before this?
Well, apparently not these two. I don't know if there were others.
Yeah, I was trying to just look in the
now this paper is
genome browser.
It's from last January.
January 2013.
Right.
Yeah. But that's
by the
okay
way,
still, right?
Oh, that's fine. That's fine. I'm pointing out, maybe there have been more since then. But in addition, it also means that I believe this is open access now on the science website.
Yeah, I think it is. I think I got it from home, in fact, yeah.
People want to go there and grab it, they can. They
so
can
the penultimate author, Lin Fa Wang, has invited me to visit his lab in Australia. And
cool.
So I'm going to do a TWiV with him this coming July. So we'll hear more about bats and genomes and this sort of thing.
Great.
Okay, so I just looked in the UCSC genome browser, and they have Microbat and Megabat. Are those going to be these kind of bats? Those genomes?
I think, the flying foxes are megabats, and the myotises are microbats, yeah, I think.
Yeah.
I know they're small.
So they did have some bat sequences. Yeah, Microbat is the Myotis lucifugus, and the Megabat is Pteropus vampirus. Okay. Vampire bat.
I think basically these two are worked on in labs, and they wanted to have their genomes, but there probably were others, yeah.
So yeah, it looks like there's at least 4 bat genome sequences now.
Okay. All right, let's do a few emails here today. Kathy, can you take the first one, please?
Scroll to Sophie.
Yeah.
Yeah. Sophie writes, "Hi, TWiVies. I have a somewhat useless follow-up concerning gemütlich or hygge in Danish. As a Dane, I consider myself somewhat of an expert, as it is the only way to get through a winter that consists of 7 hours of daylight, cold rain, sleet, etc. While the definition Rich found is completely correct, I found it somewhat lacking. Hygge is curling up under a fleece with a cup of cocoa watching your favorite movie. Hygge is the smell of home and love. Hygge is opening presents with your family. I guess it's everything nice distilled into a few hours, and it's the only way to get through a Scandinavian winter. Happy New Year. Sophie, the biotech student who wrote you a couple times long ago before I became a vet student."
Yeah. Wow, that makes it really clear, doesn't it?
That's great.
Scandinavian winter. I guess we can't complain, is that right?
Right.
No, I guess not.
We have more daylight anyway.
But apparently, I guess you'd just say that you need a hygge.
Hygge.
Right.
Very good. Alan, you're next.
Greg writes, "Your discussion of EMCV on TWiV 262 made me wonder whether that might be the reason elephants are so afraid of mice." Provides a link to a Mythbusters about whether elephants are afraid of mice. On a more serious note, I've heard it said that the mouse is a good model of the human immune system. Given that, I'm wondering if you had any comments on the work by Siak et al. Published in PNAS at the beginning of 2013. Gives a link to the paper and then quotes, "We were surprised by the poor correlation between the genomic responses in the mouse models and those responses in human injuries, especially given the worldwide prevalence of the use of mice to model human inflammation." And Rich actually sent this along. Should I go ahead and read his response?
Sure.
So Rich says, "I asked Jeff Ginsburg, who's looked at the transcriptional response to virus infections in humans, about this, and I got this response back. We have not compared the immune response, but in mouse models of H1N1 infection, the host transcriptional signature is similar to the human. We have not done extensive studies in the mouse model. We prefer the human." Okay.
So basically in this paper they do a transcriptional comparison of inflammatory conditions in mice, right? And try and see what it looks like compared to humans. And the conclusion is these results show that the genomic responses to different acute inflammatory stresses are highly similar in humans, but these are not reproduced in the current mouse models. New approaches need to be explored to improve the ways that human diseases are studied.
I don't think anybody would disagree with that last sentence.
I totally would agree. And especially something like inflammation, where lots of genes are involved, and we know that mice are inbred and humans are outbred, right?
Right.
There
are all kinds of problems with the mouse model. It's just it's better than any other model that we have access to. I mean, you can't do these studies in humans, and we've got centuries of genetics on mice, and we've got all this background on them, and they're easy to work with, and they don't eat much, and they're cheap, and
immunological reagents.
Yeah, and their immune systems for a lot of types of work that people do, they're close enough.
Mm-hmm. Yeah. Well, we did a paper on TWiM yesterday where they have a mouse model of colorectal cancer where they treat the mice with a carcinogen and an inflammatory agent, and the mice develop colon tumors. And if they deplete their microbiome with antibiotics before, they have many, many fewer tumors. And that's correlated with a specific depletion of certain types of bacteria in the gut. In humans, if you look at people with colorectal cancer, there are certainly alterations in the gut microbiome, but they're different in every set of humans that you look at.
Right.
And it's just because humans are outbred, and there's going to be lots of different ways that this can happen. So, but we still love mice. They're just not people like ferrets, you know.
Yeah.
The next one's from Jim, who says, you've mentioned the concept of funding. Yes, indeed, we have. Perhaps a tactic used by Mike with his tech show can be used to accumulate funds to help pay for the transcription work, so the folks working on it can spend more time extracting proposed episodes. And the link. Mike's tech show makes money by having people link to Amazon through his site. It seems to provide a moderate income. This link shows his affiliates, including Amazon. The link from show notes 478 shows the redirect text he discusses for about 5 minutes, starting around the 26-minute mark. Amazon for Dummies extract provides a concise explanation of the affiliate program. It appears to pay about 3% of the price paid for purchase. All your listeners do is use the link on your website to deal with Amazon. I've used Mike's link a few times and haven't seen any differences from going directly to the site. The only concerns are professional and institutional constraints you have with using the website to raise funds. Well, we do use the Amazon affiliate links. If you go to our TWiV bookstore, which is on Amazon, every pick of the week that's a book, I put there a link to, and anyone who purchases the book through Amazon supports us. But I must tell you that the most we've ever made from that is about $10 a month.
But I gather this is a much more general mechanism where people would click a link on the TWiV site.
Yeah.
Anytime they, instead of typing Amazon.com into an address bar, type TWiV.tv, click on that Amazon link, and then Amazon will detect that you came from TWiV and will kick back.
I see. Yeah, it's not specific
some
stuff.
percentage.
Yeah.
So if I'm going to go buy, I don't know, some new
Snow wobble.
Snow wobble, exactly. I would click through from TWiV and get this.
Yeah, I could put these on, that's for sure. I'll do that, no problem. Any little bit helps.
That's a good idea.
Mm-hmm.
Kathy, we're back to you.
So this is Jim again.
Yes.
Just so it isn't overlooked, a PDF version is attached. What I found interesting is the existence of an apparent group of anonymous whistleblowers who sound like trolls. So he sent this article, which is a long article from Nature in November that I hadn't seen, and I really only had time to scan it. But the gist of it is they talk about some really dogged whistleblowers, and then toward the end, they start talking about anonymous whistleblowers. So you guys probably read it in more detail.
Yeah, I think what Jim is referring to is this National Whistleblower Center in Washington, which it's a nonprofit group that exists to try to protect, I gather, whistleblowers of all sorts, whether they're in science or business or government or wherever. And he's one of the sources quoted. So I don't know that I would say that that's trolling, but unless he was referring to the anonymous, well, maybe he was referring to the anonymous one.
This article, right. He talks about 3 different kinds of people.
Yeah, okay, so Jim is probably talking about the Claire Francis phenomenon.
Mm-hmm.
Yeah, and so, people who are just looking for fraud in science, so sort of like looking for trouble or looking for discussion. So
and a lot of the anonymous complaints that are now pouring into a lot of journals are very incomplete and not really something you could follow up on. And apparently, if this is one person, he or she is a bit what's the word I'm looking for here? Crotchety about how these things get handled.
Right.
But it could just be a bunch of people using the same pseudonym who are essentially, in some cases, coming up with legitimate gripes, and in other cases, apparently trolling.
Right. So supposedly this Claire Francis has emailed about 100 different editors, according to Francis. And the executive editor of PNAS, Diane Sullenberger, says that as many as 80% of the allegations they get come from Francis.
Right.
So.
I guess this can waste a lot of time also, right?
Yes.
Right. Right.
Right. So, on the one side, you want people to feel free to speak up if they see shenanigans, but on the other side, you don't want everybody to be wasting their time on wild goose chases.
Right. They say very specifically that many of her claims did not check out. I have no problem taking time to look at an allegation, but I don't like people wasting my time, said Eric Murphy, editor-in-chief of Lipids. So,
I think it's fine if you read a paper and you see something, but if one person is doing hundreds of this, I don't get that.
That's kind of crankish.
Yeah.
Yeah.
I don't know why he or she would want to do it.
Or they.
Yeah.
Or they. Yeah, they probably.
Yeah.
What are we up to here? We are up to Marion, and that would be Alan.
Okay. Follow-up letter to TWiV 264. Contradiction and viral can become bacterial, not transubstantiation. Okay, I don't know what that was in reference to. Anyway, starting into the main letter, I was struck by the 180-degree about face in episode 264. Near the beginning of the episode, there was a question about whether the, quote, public is interested in science. All the hosts decided that the public is definitely interested in science, even tow truck drivers. However, toward the end of the episode, the discussion included unsubstantiated negative comments to the effect of 80 to 90% of the public does not know the difference between a virus and a bacterium, maybe even what a cell is. I think these kinds of discussions are mostly pointless since they're not backed by data, thus the easy turnaround. Moreover, if data exists, they're largely based on self-reporting. However, I disagree with Alan that the anti-vaccinators, the climate change deniers, the creationists are marginal. They may not be a majority, but their vociferous, willful ignorance is very dangerous, and the more noise they make, the more that neutral, less educated people or policymakers may be swayed. I don't see this as a contradiction. Yes, fringe groups can wield outsized influence, but as for the public knowledge of science, there have actually been quite a few survey studies of that show exactly these sorts of results. So it's not really occurring without data. I don't know. I think all these things are fairly part of a fairly consistent view. The public is interested in science but doesn't necessarily understand it. Mm-hmm. Continuing, secondly, regarding the issue of confusing viral and bacterial infections and treatments, I did want to point out one thing. A viral infection may predispose a person to a secondary bacterial infection. Examples include influenza and pneumonia, HIV and secondary infections, measles, also probably rhinovirus and strep throat. Good medical care might include close monitoring of viral infections for subsequent secondary infections, possibly including prophylactic antibiotics in certain cases, such as someone who has recurrent bacterial infections. Of course, more and better antivirals would be the ideal. I would agree.
And I think this is in regard to the listener who wanted to know how many people would understand the difference between antibiotics and antivirals. And I posed that question to my class at the beginning of the second lecture, and I haven't downloaded the results chart. I looked at it briefly during class, but they're not the general public, obviously.
Yeah.
Right.
So
I'm going to do that also in my first class next Wednesday. Mm-hmm. But it's closer to the general public, right?
Yeah.
Wait, yours are undergrads? I'm sorry.
Yeah, no, they are undergrads.
Yeah, so are mine. It's the same thing. Yeah.
Yeah.
Should I continue this next bit? Okay, so if this brings up the topic of antibiotic abuse and misuse, I have several comments. A, I disagree that the overuse of antibiotics causes the emergence of superbugs. This goes in the category of post hoc non propter hoc. It's correlative only. I don't think data exists to test this hypothesis. My opinion is that any use of antibiotics selects for resistance, and I doubt we can return to a no antibiotic era. We need to develop better ones and use existing ones better. I've seen quite a lot of data that I interpret to show that antibiotic overuse is a major driver of superbug evolution, and that's why these species, these strains, seem to appear first in hospitals and factory farms.
Yeah, I think an example is the emergence of totally drug-resistant TB in India, where misuse is huge, and antibiotics are misappropriated and inappropriately prescribed and so forth. Yeah.
Right. B, the ID docs need to take a lesson from HIV cocktail treatment. The multiple drugs are not used because they're stronger or because of provirus. They're used to slow replication and therefore mutation and therefore the emergence of drug resistance. If we did the same with antibacterials, i.e. simultaneously hit with multiple effective drugs, I believe we would actually decrease the emergence of resistance, but it needs to be tested. And I'm sad to report that that is actually tested every day in every major hospital of every developed country.
Multiple antibiotics?
Multiple antibiotics. If prescribing more antibiotics and multiple antibiotics would prevent or wipe out superbugs, there wouldn't be superbugs.
But isn't most therapy monotherapy?
It tends to be, but I just think I'm pretty sure that this experiment is ongoing.
Because you think a lot of docs have the incorrect impression on how multiple drugs work?
No, I don't think the doctors have an incorrect impression. I think they're trying to save individual patients. Doctors don't think the way scientists do. They think in terms of this patient needs,
I just meant with the ID docs need to take a lesson. The multiple drugs are not used because they are stronger or because of provirus. It seems to me that the implication is that the docs don't understand how multiple therapy works.
I have the feeling that ID docs would understand that. So the idea is, for the HIV drugs, you're hitting several different parts of the life cycle, and therefore reducing the chance that you're going to get a drug-resistant virus in an individual patient.
Yeah.
At least that's what I understand, so I'm not an ID doc.
Okay.
No, ID docs are my father-in-law was one. They're extremely familiar with how resistance develops and resistance management, and that's a huge topic in the field.
I mean, he was an ID doc in a major academic medical center. Do you think in smaller hospitals, private hospitals, public hospitals, it may not be universally true?
I think it's pretty broadly true. In fact, I've had this experience in talking with a pediatrician here in Western Massachusetts who was prescribing an antibiotic for my daughter and started into a spiel about, how this had to be managed carefully. And I reminded him of my background. He said, "Oh, right, I don't need to tell you this." Yeah. So this is, and that's a pediatrician. Well, infectious disease is pretty huge in pediatrics, but
Western Massachusetts is a highly developed area of the country, though, isn't it?
Yes, of course.
Okay.
Just finishing up Marion's letter, one more comment on reprogramming T cells for leukemia therapy, New England Journal of Medicine. I totally agree that this is totally cool. What my undergraduate students were more cognizant of was that it used a lentiviral vector, i.e. HIV. They thought that HIV cures leukemia. Yeah,
a lot of people get, in fact, headlines in newspapers touted this, HIV curing leukemia. It's crazy. Yes. I've seen that. All right, next one is from Jean-Michel Claverie, who writes, "Dear Vincent, thank you for your continuing interest in giant viruses. I can already tell you that the new year will not be devoid of surprises concerning them. The saga will continue." The paper should come out in early February. Fingers crossed. I will send you a proof, but you will have to respect the embargo. Of course. In the meantime, best wishes for the New Year's on behalf of myself and Chantal. So cool. Let's get more big viruses in 2014. Thank you. That's cool that he writes in. Shall we do one more round? Is that okay with everyone?
Sure. Yeah, I think we can do that.
All right, that brings us back to Kathy.
Toph, or Toph, T-O-P-H, writes, "Hey TWiV team, the temperature here in San Antonio is 12 degrees Celsius, and it was pretty sunny today. I have a couple of questions. My first is about vaccines. It has to do with the principle behind vaccines. Exposure confers immunity. Wait, Is confers the right word, Vincent? He's asking. I think confers is okay as a word.
Yes.
Yeah. With this in mind, how likely would it be, or have we seen this already, that exposure would not give immunity? So should I stop, or and should we answer that? How likely would it be, or have we seen that exposure doesn't give immunity?
Yeah, we could address it. Sure.
Yeah, there's at least cases where exposure and infection by viruses doesn't give long-term immunity. I mean, it's one of the things we've talked about with, for example, noroviruses. That's the first thing that came to mind for me.
We know the flu vaccine isn't 100% protective.
Right.
It may be that a certain fraction, I mean, for all vaccines, we know a certain fraction of recipients do not mount a protective immune response. So for
right,
sure.
and of course, there are viruses for which
Yeah.
exposure doesn't give immunity.
Yeah, exactly. So yes, it's not, I mean, it's not hugely likely, but it happens, right?
Right.
I mean, I wouldn't say
right,
it.
we should distinguish between a virus where most people get immunity from exposure versus a virus where most people don't get immunity from exposure.
Right. I mean, it depends on the virus and also the population, you know. So
Right.
I think polio vaccines have a very high protection rate, but flu vaccines don't. It also depends on how the vaccine is made. So the answer is for sure.
Right. Right.
And in cases where exposure, where you've got, say, flu vaccines, where exposure confers immunity on some people but not others, it may just be, or probably is, that we just haven't really worked out all of the right correlates for immunity. So it may, the antibodies against flu may not be the main protection mechanism.
Right.
We don't know.
But I think the point is that licensed vaccines have to do better than a certain percentage of protection, right?
Yeah.
Mm-hmm.
And, you know, so the polio vaccine is better than 90%.
Right.
And the flu vaccine isn't as good, but it's still better than 50%, I think.
Mm-hmm. Right. Okay, and he continues, we heard this thought briefly mentioned in one of the discussions about HIV vaccines and some of the problems in making HIV vaccines. I was wondering if this is a major issue. So, okay. Now, his second question. If this next question is too dumb, you don't have to read this one. Well, it's kind of over my head, but it's because I don't really understand the zombie phenomenon. So I will read it, and then I'm writing a short story as part of a writing group my friends have started, and I need a plausible zombie virus, something that replicates in the saliva and is active on the brain. I don't really want it to be the reanimation type zombie, mind you. Insertion from me. I don't know what a reanimation type zombie is. Okay. He continues. My friends know about.
It's a zombie that comes back to life, right?
Okay.
Is a zombie living?
Oh, okay. Right. So the I gather, I haven't watched a lot of zombie movies, but I think the zombie as the undead, a dead person who's raised from the dead, is wandering around as a zombie is one person, and then the other is somebody who's alive who gets some kind of a spell or virus or something that causes them to be a zombie.
Mm-hmm.
So he's not interested in the reanimation type of zombie, but the infectious type of zombie.
So a virus that would turn someone into a zombie.
Right.
Which is what?
Rabies?
Yeah.
Well, I mean, what would, like, if I became a zombie, what would I do differently?
Oh, how would we know?
Yeah. You wouldn't know because I already am a zombie.
And no, I think a common theme is they go around trying to find uninfected people and eat brains and.
All right. Can someone write a concise explanation of this zombie business to us?
Right. What symptoms are we looking for here?
Okay. Anyway, I'll continue. My friends know about my interest in viruses, and they know I listen to TWiV, although I don't think they themselves are listeners. It was kind of their challenge for me to come up with something interesting. I thought Dixon or Rich might enjoy throwing around some ideas.
Neither one is here.
Right. Okay, here is his stab at it. An endogenized pallidna-like virus with different encapsulated DNA strands that work on separate parts of the body to produce the symptoms we need. It would be vertically transmitted, and it would break free of the genome when triggered by a chemical attack. I wonder if he really means vertically transmitted, from parent to child?
Oh,
Or does
so he
hoard?
means something that's been in the DNA the whole time.
Yeah.
Oh, he does mean that. Okay.
That would be reactivated under some condition of the plot of the story. Like a
Right.
chemical
Could
attack.
be reactivated when an aphid bites you.
Yes.
Right.
That would be
Right.
good.
I need it to be as plausible as possible, and if this becomes an ongoing thing, I want to use it as a platform to explain what an endogenous virus is, what real virus does, the difference between mortality rate and case fatality ratio, and eventually zoonosis, retroviruses, vaccine types, and, well, whatever I can work in. I'll send you the link to the Tumblr when I've written it. And if you guys have any other ideas or further ideas, please let me know. Thank you. Thanks, guys and gal, if you're there. Maybe I'll name a few characters after y'all. P.S. I wanted to clarify, I know vaccines work. What I was wondering is if we've ever run into a virus that we keep catching, something that is still infectious to us, even if we produce a complete immune response.
Well, as you said, Noro's keep
Noro.
getting infected, but that's probably because the antibody levels wane, right? Or there's there's antigenic variation or something like that.
Right, but you produce a complete immune response, but you still get the virus again later.
Mm-hmm.
Right.
I mean, none of your big infections, measles, mumps, rubella, polio, flu, I think fit into that category, right? A good immune response, but still get infected.
No, the good immune response is kind of defined by not getting infected again.
Yeah.
Right.
Of course, all immunity tends to wane with age.
Yeah, for sure.
Right.
So what do you think of this his virus here?
Well,
Okay.
again, given that I still don't really know what these zombie things are, I was thinking about human cytomegalovirus because I know it's found in the saliva, which makes me think maybe it's latent or replicates in salivary cells. So maybe he'd be happy with something that, could be latent and then reactivated by he wanted a chemical or something like that, so it would break free of the genome.
Actually, herpes virus would be a really good candidate for that.
Well, the problem is most of us have multiple herpes viruses, right? Latently, and we don't.
Yeah, but
don't.
that makes it an even better candidate, I think, depending on what the plot is supposed to be, because something happens and then all of a sudden most of the population is running around doing zombie things.
Right.
And then your characters have to find their way through this this apocalyptic landscape of zombies, right?
So you're saying some certain trigger would make it
would reactivate this virus, this CMV-like maybe virus that was lying there dormant, and you could then talk about how that happened.
But why would it be different from a natural reactivation of CMV, where you get some mild
Well, it
symptoms?
would have to be a different virus besides CMV.
Oh, he's got to design a virus to do this. I see.
Right. So I think
Right.
he wants to design a virus that's credible, but would produce these fictitious.
Yeah, it doesn't have to be endogenized. I guess he wants to be transmitted in the germline, because that's what endogenized means, right?
Right.
Vertically transmitted, yeah. And so CMV doesn't fit that. But, HHV6 seems to endogenize, right? So in a certain percentage of infected people.
Right.
Into the telomere, and it's passed on in families. So that's
Or
a
you could have
possibility.
something that's not necessarily endogenized, but it's harmless, so it's widely spread, and then in response to some sort of environmental trigger, that changes something about the virus.
Yeah, so like we're all infected with these single-stranded DNA viruses.
Yeah.
And if you could figure out how to make them be nasty when they're
right. You
induced.
want to evilize the whatever common virus is already spreading.
Well,
Okay.
There are some ideas.
there's some ideas, and if listeners have ideas for TOF, tough. All right, Alan, you're next.
John writes, it's -18 outside as I risk creating an infinite loop by writing an email in response to an all-email episode. I hope that's Celsius. I liked Rich Condit's comments about 1984, doublethink, and critical thinking in TWiV 253. First, my own observations, then I'll explain how this relates to TWiV. I listened to episode 253 around the time the U.S. Department of Transportation released its annual poll of drivers. As usual, most drivers said that speeding is bad and they exceed speed limits anyway. That's doublethink. Critical thinking was conspicuously lacking as reporters cut and pasted speed is bad press releases as if they were a sound basis for policy. Apply reductio ad absurdum. If speed is bad is the basis for policy, we should freeze in place forevermore. There's more to the story. On TWiV, I hear a lot of vaccines are good generalities when there's more to the story. In episode 265, you mentioned the HPV vaccine again. I'll accept that it's safe and effective at preventing HPV infection. Now, who should get it? Prevents cancer does not answer that question. Apply reductio ad absurdum, the honest claim is probably reduces risk of cancer, and the vaccine costs money to make. What if it cost $1 billion to reduce one person's risk of cancer by 1%? There must be some limit. We can't make sound policy without a cost-benefit analysis. I hope the TWiV crew can fill in some numbers, such as the HPV vaccine costs X dollars per person, each vaccination saves Y lives and prevents Z dollars in future medical costs. On the medical side, there's some guesswork involved. Imagine an XMRV vaccine to prevent prostate cancer before we learned that XMRV doesn't cause prostate cancer. I gather the HPV cancer causal link has more solid evidence. Oh yeah. But nobody has done 40 years of trials to quantify long-term effectiveness. On the economic side, the value of a life is reasonably well understood. I recommend the 22 February 2013 Science Magazine podcast. They discuss cost-effectiveness of HIV treatment in Africa. A key measure is the ratio between cost per year of life saved and per capita GDP. It gives a link to the article. I found partial numbers for the HPV vaccine. My impression is it is moderately cost effective when purchased in bulk for mass vaccination campaigns. From a business perspective, that makes sense. To maximize profit, the price of a drug should be as high as possible without making the cure worse than the disease. But I don't have accurate numbers to base that conclusion on. Maybe you do. Yes, thanks. Glad you asked. Rich and I apparently went and essentially PubMeded up similar stuff at the same time, and he also has some additional comments here, so I'll start with his. Rich cites a CDC study, Cost Effectiveness of Human Papillomavirus Vaccination in the United States, and quoting it, the estimated cost per quality-adjusted life year, QALY, gained by vaccination in the context of current cervical cancer screening practices in the United States ranged from $3,906 to $14,723, and that's in 2005 dollars. Rich says, I spoke with my nephew-in-law, who's a health economist, and I can explain this statement. Importantly, any intervention that is valued at less than $50,000 per QALY is considered cost-effective. So yes, HPV vaccination is judged to be cost-effective. See also, what is the price of life and why doesn't it increase with inflation? These authors suggest that the threshold should be $200,000 and continuously adjusted for inflation and other factors. And I also came up with another study, which is health and economic outcomes of HPV 16 and 18 vaccination in 72 countries that are eligible for the Global Vaccine Initiative. So that paper shows HPV vaccination would also be cost effective in poor countries if we can bring the cost per dose down to $10 or so. That paper used disability-adjusted life years, which are analogous to QALYs. And I would just add, as a footnote here, this type of calculation is done routinely, frequently, and repeatedly for every vaccine and many drugs, even before they get to market.
Yeah, because the companies wouldn't make them if they weren't.
Companies won't make them if they're not cost effective, and government-funded government agencies and insurance companies won't pay for them if they're not cost effective.
Right.
So vaccine makers are keenly aware of this cost effectiveness issue. There was some controversy about the HPV vaccines because they are, in vaccine terms, they're significantly more expensive than the usual, but they are still very, very cost effective when you express them in adjusted life years. So yes, this calculation is done, and this calculation is done again and again. The CDC is particularly an avid user of dailies and QALYs.
On the question of whether the HPV cancer causal link has solid evidence, how about Nobel Prize winning after, 30, 40 years of experiments by Zurhausen, and others? Yeah, that's...
and we're down to mechanistic details on which genes cause which effects.
Right.
It's...
But so the point that, nobody's done 40 years of trials to quantify the long-term effectiveness of this vaccine, that's absolutely true. But
it's ongoing. You can't know the answer until you do it. You have to do it.
It is ongoing, and the preliminary data are extremely encouraging.
Right.
HPV infection rates, and you look at seroconversion rates, and you can follow this in a variety of ways. And for young women who've been vaccinated, they're lower. The thing works.
Just saw a paper on the use of HPV in the U.S. I think I'll dig it up next time and we can mention it. But it's not as high as you would like it to be.
Right.
A lot of people are not giving it to their children. All right, the last one for today is from Bill, who writes, I became aware of TWiV and TWiP about 3 weeks ago and have listened to each new episode as they occur, and I am also playing catch-up via the RSS feed, and I estimate that will take me about a year. I find it very interesting in its own right. I'm a retired chemical engineer, and it is by far preferable to the banal talk shows and music as I perform my exercise routines 4 to 5 times per week. I am not studying this matter, but it occurred to me that all manner of coursework could be prepared in this manner as a supplement to classes and lab time, so the students could use dead time more productively, since adding this audio stream to the class and lab streams could cement the knowledge into long-term memory more effectively.
Hear, hear.
Yeah.
I'm trying to get my class students to listen to the TWiV 101s, and I don't know that they're doing it, but... Yeah, the virology 101s.
I agree. That's why we do this. And audio is a good way to reinforce. And, of course, this is the flipped classroom concept where you let students listen ahead of time and then you talk about it later.
Yep.
I've listened to you and others on TWiV complain about the dumbass vaccination deniers, and that made me ask, is a denied vaccination and pre-existing condition, and as such, can insurers deny coverage for any disease that could have been vaccinated against? What if the insurer has a questionnaire and asks if you had vaccinations for 1, 2, 3 illnesses, and as you answer, so is your rate determined? Non-vaccinators will pay a much higher rate, which is reasonable since their refusal to vaccinate reduces the herd immunity of all of us and thus increases our costs. If people say they have vaccinated their kids to get the lower rate and get an illness, and it is found that they lied, they bear true costs. Of course, these deniers all want their cake and they want to eat it too. Has this scenario ever played out in the new Obama system, and if so, how have the insurers ruled? I don't know.
Yeah.
I don't think you could base insurance on vaccination or not, right? Because it's not 100% protective, right?
Right.
Well, it's not 100% protective, but if you can base insurance rates on whether or not you smoke and whether you're obese and other risk factors, not being vaccinated against, say, measles is a huge risk factor.
Yeah. So why isn't that happening?
I don't know. The only thing I can think of is political pressure from a very vocal minority. Rich actually provided a link. Rich seems to have answered half these emails for us.
Right.
He provides a link to the Wikipedia page about vaccination policy in different countries. And I think we've even talked about some of this. There was, might have been Australia. There was some place where they were going to impose a fine for people not getting vaccinated because these people are externalizing a cost on society, and so they ought to pay for it.
Yeah, we did talk about that a while ago.
And that boosts vaccination rates, of course. The way we've typically handled it in this country is, at least on a state-by-state basis, school systems have requirements for vaccination for kids.
Right.
And so since most of the vaccines are for kids, my daughter can't go to the public schools if she hasn't had her shots. Simple as that.
Well, you could have a medical or religious objection, right?
In Massachusetts, you can only have a medical objection.
Oh, cool.
And they're serious about it. You've got to get a pediatrician to to sign saying what the objection is. So, but other states are more lenient about that.
At this Wikipedia site, if you go about a third of the way down, they list things country by country.
Mm-hmm.
So, for instance, in Australia, the federal government made certain benefits dependent on vaccination.
That was it. Yes.
Yep.
So, and then they go through and list a number of other countries. So it's worth taking a look at.
Yeah, take a look. Alan, are there any other states that will only take a medical exemption for not getting vaccinated?
I certainly hope so. I haven't. That's just the only reason I know this about Massachusetts is personal experience, because we got how to enroll your child in school, and I saw that and I said good.
All right, let's do some picks of the week. Alan, what do you have?
Well, I have the item that I mentioned at the beginning of the program, which is it's a wireless thermometer. It's made by a company called Ambient Weather, which is kind of a funny name because I thought weather was always ambient. But and this was a Christmas gift. It's not one of these full-fledged weather stations with some people go all out. They get the wind speed gauge and the rain gauge and everything, but those require maintenance. You take something that's about the size of a deck of cards and you mount it someplace outdoors that doesn't get sunlight. So I attached it to the north side of my house, which is the outside of my garage. That's always shaded. Of course, if the sun were shining on it, it would drive the thermometer nuts. And then the inside part of it is this thing that's I don't know. I guess it's about oh gosh, I'm gonna step away from metric now. It's about 6 inches tall. So that's what.
Pulling out my metric ruler
15,
here, yeah.
20 centimeters, something like that,
15.
by about 10 centimeters wide. And it stands up like a picture frame, or you can hang it on the wall, and it gives you the indoor and outdoor temperatures, the indoor and outdoor humidity, because there's a hygrometer built in, the barometric pressure. It'll tell you the trend of the barometric pressure, and it's got little icons based on whether the barometer is rising or falling. It'll tell you is your weather moving from sunny to cloudy or cloudy to sunny, or you're probably going to get rain or something like that.
Cool.
Mm-hmm.
Unfortunately, I couldn't use it here in my office because I couldn't put it outside.
No, you couldn't get to the outside of the building.
These are great for homes. You going to get one, Kathy? You wanted 3 weeks of purchases in a row.
I don't know. I did get the virus wrapping paper, though, part of the wrap project. So I did get that. So I already have a thermometer at home that tells me the outdoor temperature from inside my house, and that's probably good enough. So.
I have one. The problem is the north side of my house is my garage, so I couldn't if I put a thermometer outside the window there, it wouldn't be useful.
Right.
So I went and I got one when we first moved into this house. I picked one up at the hardware store, and I mounted it outside the kitchen window, and I didn't really think about where that was. It faces due west, so it's reliable. It's kind of sort of reliable for the first third of the day, and then it just goes bonkers.
Yeah.
Well, that's what you used at the top of the show, right?
Mm-hmm.
Yes.
Cool.
Yes.
All right.
Yeah, very nice.
Kathy, what do you have for us?
Okay, I have a series of links. Sometime in the fall, Jean-Luc Demont came here and gave us two seminars up on the engineering campus, and people from my lab went to it and came back saying he was fantastic. And one person took notes, but there were links to a couple of his YouTube videos, one on making effective slides, one on communicating science, and there are things on his website. There's a PDF you can download, and it's just really he reinforces a number of things that I've figured out by trial and error in terms of presentation, but he's got a number of other good points that I thought were really worth taking a look at. And I really just kind of it was one Friday night, and I kind of went down the rabbit hole of watching almost everything. And there's a whole nature site called Citable, Scitable, and part of it is a whole set of links about English communication for scientists. So writing, how to write correspondence, how to give oral presentations, how to interact during meetings, communicating in the classroom. And he's part of the giving oral presentations thing, so that was the connection there. But they're really well worth looking at. And once you see his slides and his style, you might or might not want to embrace it, but it certainly makes you more conscious of other people's style slides that are maybe the antithesis of what he's recommending.
Yes.
So
I think you're right. We have picked this up as we went along, right? Most of this stuff, trial and error.
Right.
But,
But he, for instance, I mean, just to tell you one that was kind of earth-shattering for your, the tendency now is to put the conclusion at the top of a slide and centered. And his thing is, no, put it at the left edge so that the first thing that people's eyes see is that left edge. And you do that for every slide. And he doesn't use PowerPoint or Keynote. I forget what, it's some kind of advanced computer thing, Alan, that you would recognize. But that makes it very amenable to a template and all your things always ending up in the exact same part of the slide. So he has some, they might sound kind of rigid, but if you're just starting from scratch for a presentation, it might be worth thinking about some of these things.
So you left Justify, and that makes all the difference in the world.
Yeah, because I find too that even if somebody has something at the top of the slide, I kind of jump down to the data or the body of the slide. But if you have something at the top left, that's where your eye goes first.
Right.
I don't know. So.
Yeah, I do put centered text at the top, but more and more I'm getting rid of the text and I just put a picture on and talk about it. I think that makes them look at the picture and listen to you.
Yes.
I have a interesting article on rethinking the airline boarding pass. Now, last year I flew over 60,000 miles, all on the same airline, United, and every time I looked at a boarding pass, it always took me at least a minute to figure out where and when I was going, because boarding passes suck. Plus, if you put them in your passport, the ends stick out, and then if you put that in your pocket, they get bent, and it's a mess when you think about it. So this fellow has a great solution. He's just redesigned it to be really clear, could be printed black and white, and if you put it in your passport, you fold the top over and it fits perfectly. And then the top sticks out and you can see your flight and gate number. So I think this is really cool. Now you may ask, why don't you just use your smartphone to show the boarding pass, right? So I've done that a lot, and there have been a few times when it didn't work.
Right.
Bad reception or bad whatever. So I always print out a boarding pass, either at home or at the airport, because I don't trust that my phone is going to work every time. And I've sat next to many people who fly a lot more than me, and they always say, I never use the phone. I always print out a boarding pass because it works.
I've been stuck behind people using their phone several times, and it gums things up.
Yeah, so I think the technology isn't there that it's perfect yet, if it ever will be. So in the meantime, I wish the airlines would check this out, and it's a really nice design.
Yeah.
Yeah, especially if you go to the very end of the link, the site, he's got it diagrammed for how it'll be the same for different airlines, but they have their different colors and their logos and
Right.
Yeah, this is somebody who just thinks about design, obviously, and did a good job, and I think he's happy if the airlines just take his design and do it. So let's see who does it.
Yeah.
I have a couple of listener picks. One from Hannah. Dear TWiV, I was disappointed that you did not do any picks of the week on this episode. This is our first of 2 episodes at University of Wisconsin-Madison. I'm sending along a link to the book that Dave mentioned, Practical Computing for Biologists. Perhaps you could add it to the links for the episode and add it to the master link list of weekly picks. So we will make it your pick. And I have actually bought the book. It's very good. It is designed for scientists, how you use computing to make everything that you do easier. So I highly recommend it.
Cool.
And you can put it on the Amazon bookshelf, too.
I will.
The TWiV bookshelf.
And one more from Stephen, who writes, I wanted to send this listener pick of the week now so I could mention the weather here in the St. Louis, Missouri area. It's 12 Fahrenheit with -3 Fahrenheit wind chill. There's about 9 inches of snow on the ground, and it's still snowing. Forecast low for tonight is -10 Fahrenheit. And the video he sends is 14 years of U.S. weather. May 2nd, 1997 to December 31st, 2011. It's just sort of a quick time-lapse movement of the weather patterns over the U.S.
Yeah, I put it on my other monitor and just started it going, and I stopped it after about 8 minutes because I realized I was going to miss a whole bunch. But
it's a half hour.
Yeah, it's a half hour long.
It's kind of like Florida, New Jersey in 156 seconds.
Yeah, that's right.
Yeah.
All right. That's TWiV 268. And you can find us, of course, as always, on iTunes at twiv.tv. And you can send your questions and comments to TWiV at twiv.tv. And, of course, go on over to iTunes and give us stars and comments if you haven't already to keep us more visible and let more people find us. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was a lot of fun. And I learned a lot about these viruses and bats. It's cool.
Plant viruses and bats. What a combination. Mm-hmm. Alan Dove is at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
And enjoy your new thermometer. Well, it's more than a thermometer, right?
Yes.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.